this is important chapter also. So when we manufacture the drug, the one of the important part is decomposition of the drug, because when we finish the manufacturing of the drug, it starts to decompose, isn't it? But the rate is different. The rate of decomposition is different, but the decomposition starts right from the manufacturing until it reaches to the patient where it is eaten, isn't it? So the decomposition process is very important. If the decomposition is very quick, then when the drug reaches to the patient at that time, very small amount will remain, and that may not be efficient for fighting against the disease. So every time when there is manufacturing, where there is every time when we finish the manufacturing process of drug, then the degradation rate is evaluated. So it is one of the important part of the manufacturing of drug. So there are three major types of degradation or decomposition of drug: physical, chemical, and microbiological. Let's talk about physical decomposition. Physical decomposition, the first one is polymorphs, because the drug molecules may form different polymorphs when they come in contact with other substances, or when they come in contact with UV radiation, or when they come with contact with light, or when different factors may affect, but different polymorphs may form. We know that the polymorphs has different physical and chemical properties, like solubility, dissociation rate, isn't it? Like efficacy. So the formation of polymorphs will decrease the activity of the, or decrease the efficacy of the formulation. The different factors affect the polymorphs, like temperature, pressure, relative humidity, drying, granulation, milling, and compression. So the polymorphs have different solubility, dissociation rate, and melting point, and this will affect the efficacy of the whole formulation. Hygroscopy or absorption of water. So when absorption of water takes place, then the weight of the formulation will increase automatically because the addition of water. And similarly, the addition of water will dilute the substance, isn't it? When the substance is diluted, then there will be the decrease in potency. And in case of gelatin capsule, when they absorb the water, they become sticky, and there is high chance of microbial contamination. And the factor for physical decomposition is evaporation. So when the formulation loses the water after evaporation, then the concentration of the formulation will increase. For example, if you have a solution of antibiotics, isn't it, a solution of antibiotics for pediatric use, so when the evaporation happens, then the concentration of the antibiotics will increase. What will happen if concentration increase? It will try to, the antibiotic will try to crystallize, it will try to sediment, and ultimately it will affect the potency of the formulation. If it crystallizes out, then there will be very small amount of drop in the solution, and it will have very less potency. And this is the temperature. Temperature, that is increase in temperature, will affect this whole process. The temperature will affect the polymorphs also, the absorption of water also, and this evaporation of water and other oxidation reduction reaction. The temperature is very important part for degradation for physical degradation. Increase in temperature decreases the thermolabile ingredients. It also enhances the both chemical and physical degradation process. Now, next is chemical decomposition. Chemical decomposition means the drug will react with different substances to give different products, and ultimately there will be less amount of active ingredients, and that finally reduces the potency of the drug formulation. Or sometimes some toxic products may form, and it will also hamper the health of the patient. What are the major chemical reactions that we study in chemical decomposition? The first one is hydrolysis. It is one of the most common reaction, and it is mainly due to the reaction with water, because this water is available everywhere. It is available as moisture in air, so there is very high chance of hydrolysis degradation in every formulation. Every type of formulation, there is high chance of degradation through hydrolysis. Drugs with functional group like ester, amide, lactams, etc. are very highly susceptible for hydrolysis. This type of drugs are very prone to degrade due to hydrolysis. In case of ester, we have aspirin, cocaine. Amides, we have acetaminophen, chloramphenicol. In case of lactones, we have spironolactones, pilocarpine. And in case of lactams, we have antibiotics like penicillin, cephalosporin. So another is oxidation. Oxidation is another the pharmaceutical components. The active ingredients are very prone to oxidize and go to oxidation degradation. So in this type of what happens in oxidative degradation? Actually, the drug will react with oxygen, and what will happen? There will increase in number of CO bonds, and there will decrease in number of CH bonds. There are certain types of drugs which are very much susceptible for oxidative degradation. For example, phenols like morphines, and the drugs in catecholamine groups like adrenaline or adrenaline, they are also very high chance of oxidative degradation. And similarly, polyunsaturated compounds are also more likely to oxidize. What kind of compounds fall in this group? Like oils, fats, steroids, fat-soluble vitamins like vitamin A, vitamin E. And what are the major reactions in this oxidative degradation? The major reactions are nucleophilic and electrophilic oxidation, and electrotransport, that is initiated by metals such as copper, and auto-oxidation, that is the molecule itself oxidizes, that the molecule itself undergo the reaction, that is auto-oxidation. Here I have written the example of nucleophilic oxidation. In this reaction, you can see this molecule is reacting with hydrogen peroxides and forming this type of compound. This type of degradation is nucleophilic oxidation, which is mediated by peroxide. Now I will be talking a little bit more about this auto-oxidation. What is auto-oxidation? In this auto-oxidation, the compound itself undergo oxidation into oxidation, and it will form the reactive free radicals. The mechanism of auto-oxidation is mediated by formation of free radicals, and this free radical will form chain reaction. The auto-oxidation generate the free radicals, which in turn play a role in chain reaction. The free radical chain reaction proceeds through three major steps. What are the three steps? That is initiation. First of all, the homolytic fission occurs due to the external factors like sunlight, UV, or peroxides, so to produce free radicals. For example, if this is drug molecule, then it will form free radicals. Then the second stage is propagation. These free radicals will react with another compounds or react with oxygens to give more free radicals. For example, this free radical will combine with oxygen, molecular oxygen, to give peroxide free radical. Again, this compound will react with another drug molecule isn't it? To form hydrogen peroxide form of drug and another drug, free radical of drug. And termination step is the reactive oxygen, the reactive free radicals are consumed but not generated. This free radicals of these two drug molecule will form this peroxide of drug, and two drug molecule will combine to form one drug molecule having complex of these drug molecules. So ultimately, the drug is being degraded. So in this way, the oxidation of drug, auto-oxidation of drug molecule will happen. So what kind of molecules are prone to auto-oxidation? Let's see here. Generally, this ether form of drug molecules are very prone to auto-oxidation. Drugs that are susceptible to oxidation to, what is this? Carbon hydrogen bond include susceptible for oxidation of carbon hydrogen bond include ethers, aliphatic amines, and aldehydes. Let's see how this ether and aldehyde and this amines are changed to free radical. For example, if this diethyl ether, this CS bond will break. That is, homolytic fusion occurs, and it will form free radical. The hydrogen radical and this radical of this diethyl ether, and this will react to oxygen to form different radicals. In this way, the CS bond of amine will give radical. These are the example of auto-oxidation of this molecule. Similarly, phenol, as I mentioned earlier, phenol are also very prone to auto-oxidation because the OS bond of the phenol is very likely to break to form radicals. For example, this OS bond, this is phenoxy radical will form, giving this hydrogen radical also. And this amine, this NS bond is also very prone to form free radical. So oxygen hydrogen and nitrogen hydrogen bond are cleaved to give the free radicals. Now the another type of chemical decomposition is photolysis or photodegradation. As you know that if the drug molecules, if the formulation is not covered in protective materials, then the sunlight or UV light from the sun may degrade the drug. And some drugs are very susceptible to sunlight and UV radiation. And these drugs undergo decomposition rapidly when they come in contact with UV light. So such type of drugs should be packaged in very protective materials which block the passage of this sunlight and UV light. The UV light causes more damage than other lights. Photodecomposition involves oxidation mechanism, although other like polymerization or ring opening may occur. Once initiated, the reaction can progress in the absence of light in chain reaction. It generally occurs during the storage of the product. In susceptible compounds, photodegradation creates free radical intermediates which can initiate chain reaction. To avoid photochemical reaction, photolabile formulation are packaged in call-out container. Nifedipine, chlorothalidone, acetazolamide, retinol, riboflavin, furosemide, and phenothiazines are very liable to photodegradation. The addition of antioxidants like sodium thiosulfate or sodium metasulfite hinders the photodegradation. So, the main factor for photodegradation is UV light. And photodegradation is that type of degradation in which once it is initiated, then it can happen in the absence of light also. So once the reaction is initiated, then the degradation is continued. So it generally happens during the time of storage. So how can we avoid this photochemical reaction? By using suitable packaging material, that is colored packaging material. And some of the drugs, they are very susceptible to photodegradation. So here are the example of those drugs. Now another type of chemical degradation is polymerization. What is polymerization? It is a process in which the drug molecules combine to form large molecule. They react each other and they form a large polymer. This is the process by which two or more molecules, identical molecules, combine together to form a much larger and more complex molecule. The reactant are called monomers and the product are called polymers. Example, aminopenicillin. Another is racemization. What is racemization? It is the conversion of one type of enantiomer to another type of enantiomer, isn't it? Plus enantiomer to minus enantiomer. It is a reversible conversion between optical isomers, which are also called as enantiomers. In the formulation, the efforts are made to use potent and less toxic enantiomers. However, due to racemization, both the potent and less potent or less toxic and more toxic tends to become equal after racemization. Thalidomide has two enantiomer, or thalidomide causes birth defect, while S-thalidomide is active against morning sickness. Other drugs which are prone to degrade due to racemization are penicillin, cephalosporin, benzodiazepines. First of all, we know that enantiomer. Every enantiomer, every drug have different enantiomers, isn't it? One enantiomer may have very active form and one enantiomer may have inactive form. So while making drug formulation, we use the active enantiomer, isn't it? We try to increase the percentage of active enantiomer, like we can use 90%, 99%. So we try to increase the amount of that enantiomer which are very active, which are less toxic. So what happens when we during the degradation of drug by racemization, those active enantiomer will form inactive enantiomer, and we know that racemization is a process in which both enantiomer are in equilibrium. At the beginning, we used the higher amount of one enantiomer, but due to degradation, that higher amount will decrease and they will form another enantiomer, and they will when the racemization occurs, then both type of enantiomer will be present. So when both type of enantiomer are present, then the overall what we can see, the active component has decreased to half of its original amount. So ultimately the activity of the formulation will decrease. Sometimes one enantiomer is non-toxic, the another enantiomer is toxic. So when initially we use non-toxic enantiomer, but due to the racemization process, the non-toxic enantiomer will form toxic enantiomer. So after a long time, due to the racemization, the toxic enantiomer will also increase in amount. So ultimately, the formulation will be toxic. At the initial time, there was higher amount of non-toxic enantiomer, but due to the racemization process, the number of toxic enantiomer will increase rapidly. Then ultimately, it will make the drug into toxic form. So this racemization process is also, this type of degradation is also very important. So this will make ultimately the drug less efficient, more toxic. So for in case of this third part, microbial decomposition, I have given this topic as assignment. I hope you all are doing the assignment of this thing. Now the another part is accelerated stability testing. After the manufacturing of the drug, we have to check the stability of the formulation. We have to estimate how long this drug can be stored in a room environment, that we call that we also call as a shelf life. What is the shelf life of the drug? So that shelf life can be evaluated by different processes. One of them is accelerated stability analysis. So before entering about accelerated stability analysis, let's know about what is stability of the drug formulation. The stability of a particular pharmaceutical preparation is the capacity of the formulation to remain. Capacity of the specific capability of the formulation in a specific packaging system to remain within its physical, chemical, microbiological, therapeutic, and toxicological specification throughout the shelf life. The stability means the capability of the drug formulation to remain in a specific condition throughout the shelf life. The therapeutic capacity should not change. The toxicological conditions should not change. The physical and chemical state should not change throughout the shelf life. So that capacity, that capability is called stability. In short, we can simplify it as the capacity or strength of the pharmaceutical product to remain same properties and characteristics that is possessed at the time of manufacturing. So what was the strength, what is the capacity at the time of manufacturing should remain until the time of the shelf life. Now, what is stability analysis? Stability analysis is one of the important part of manufacturing process of pharmaceutical substance. Stability study can be done to predict the shelf life, expiration date of the product. So stability analysis is the analysis to find the shelf life of the drug product, expiration date of the drug product. There are two ways to find the expiration date or shelf life. One is real-time stability study. This process is very lengthy. It takes very long time. It may take several months to years to evaluate the lifetime of the product. Because in this real-time stability study, we check the concentration of the drug for several years, how it changes until it completely degrades. We check the time. So it will take months to years to completely degrade the drug. And the next one is accelerated stability testing. It is a type of modified method in which we use certain methods which helps to degrade the drug very quickly. For example, we use here we will try to evaluate the expiry date or shelf life in very short time. So overall, what will be the benefit of this stability testing? What are the advantages from that we get from this stability testing? It provides evidence about how the quality of the drug product varies with time. It helps to estimate or establish the shelf life of the drug product formulation. It gives idea about required storage condition for a particular drug condition. And it also helps to determine the container closure system suitability. So it means that if the substance is highly sensitive to temperature or not, or highly sensitive to light or not, highly sensitive to moisture or not. So from that we can decide which container is suitable for the substance. It is also a legal requirement. So until we do the stability test, we cannot sell the product, we cannot bring the drug into market. So it is also important legal requirement for every pharmaceutical company. Now, what is accelerated stability analysis? It is a kind of stability analysis which is done to predict the shelf life or of the product by accelerating the rate of the decomposition, preferably by increasing the temperature of the reaction conditions. अब यो कस्तो एक्सिलेटेड स्टेबिलिटी अलि नेपालीमा भन्छौँ है यो कस्तो हुन्छ अब जस्तै अहिले हामीले दुईटा हेर्यौं हैन के हेर्यौं एउटा चाहिँ रियल टाइम हेर्यौं एउटा चाहिँ एक्सिलेटेड स्टेबिलिटी हेर्यौं रियल टाइम कस्तो हुन्छ भने त्यो एक्चुअल ड्रग कसरी डिग्रेड भइरहेछ भन्ने कुरा स्टडी गरिन्छ जस्तै त्यसमा चाहिँ हामीले एकदम वेट गरिन्छ त्यो ड्रग चाहिँ त्यसको जस्तै हाम्रो ड्रग चाहिँ सुरुमा 100% ड्रग छ भने त्यो 100% ड्रग डिग्रेड भएर 90% अथवा 100% बाट 50%, 40%, 60 के अरे सरी 100% बाट 90%, 80%, 70%, 50%, 40% यसै ड्रग यसरी घट्दै जान्छ त्यो रुम टेम्परेचरमा हैन रुम टेम्परेचर जसले हामीले स्टोर गर्ने रुम टेम्परेचर त्यो टेम्परेचर कसरी घट्दै जान्छ भनेर हामीले चेक गरिन्छ अब त्यो घट्दै जानलाई एकदम लङ टाइम लाग्छ हैन किन हामीले वेट गर्न पर्यो अब कुनै पनि ड्रग 100% बाट 50% आफ्नै रुम टेम्परेचर घट्दै जान्छ त एकदम लङ टाइम लाग्छ मैले वेट गर्न पर्यो त्यसलाई चाहिँ 1 वर्षदेखि 2 वर्ष 5 वर्ष पनि लाग्छ हैन त्यसरी हामीले चाहिँ वेट गरेर त भएन त्यसरी हामीले वेट गर्न थाल्यो भने त हामीले ड्रग टाइममा निकाल्न पाउँदैनौं त्यसको साथमा चाहिँ हामीले के युज गरिन्छ एक्सिलेटेड अब हामीले के गरिन्छ हामीले चाहिँ त्यो ड्रगलाई चाहिँ एकदम हाई टेम्परेचरमा राखिन्छ हाई टेम्परेचरमा राखेपछि त्यो ड्रग सुरुमा 100% थियो भने अब त्यो ड्रग हाई टेम्परेचरमा चाहिँ डाइरेक्ट 50 40 30 यसरी घट्दै जान्छ ड्रगको कन्सन्ट्रेसन त्यस आधारमा चाहिँ हामीले चाहिँ त्यो ड्रग कसरी डिग्रेड भइरहेछ भने एउटा रेट कन्स्टेन्ट निकालिन्छ त्यसबाट चाहिँ अनि हामीले चाहिँ रुम टेम्परेचर चाहिँ त्यो कसरी डिग्रेड हुन्छ त भन्ने एउटा रेट निकाल्न सकिन्छ यो प्रोसेसलाई हामीले चाहिँ एक्सिलेटेड स्टेबिलिटी टेस्टिङ भनिन्छ यसमा चाहिँ हामीले चाहिँ टेम्परेचर चाहिँ रेज गरेर ड्रगको डिकम्पोजिसन रेट चाहिँ एकदम फास्ट गरिन्छ अब रियल टाइममा चाहिँ ड्रगको डिकम्पोजिसन रेट चाहिँ स्लो भएको हुनाले एकदम लङ टाइम लाग्छ चेक गर्नलाई अब यसमा चाहिँ के हुन्छ एकदम हाई टेम्परेचरमा ड्रगलाई राखेपछि त्यसको डिग्रेडेसन रेट चाहिँ एकदम फास्ट हुन्छ र हामीले चाहिँ अब 100% ड्रगबाट 40% ड्रगमा आउने टाइम कति लाग्यो भनेर एउटा डिग्रेडेसन रेट त्यो टेम्परेचरमा कति छ त्यसको डिग्रेडेसन रेट 70 डिग्री सेल्सियसमा हैन 40 डिग्री सेल्सियस के अरे 60 डिग्री सेल्सियसमा 100 डिग्री सेल्सियसमा यस्तो टेम्परेचरमा चाहिँ कति हुन्छ त्यसको डिग्रेडेसन रेट कस्तो छ हामीले निकालिन्छ अनि त्यसबाट चाहिँ हामीले क्याल्कुलेट गरिन्छ here the reaction kinetics is enhanced so the degradation of the drug is fast and reaction kinetics चाहिँ हामीले इन्क्रिज गरिन्छ kinetic degradation चाहिँ fast हुन्छ that's why using different advanced techniques of this kinetics we can predict the shelf life of the different dosage form within a month based on the result of accelerated stability analysis. Accelerated stability testing is a modified method which we use to quickly estimate the expiration date or lifespan of the drug product when real-time stability study is unavailable. Because real-time stability, it takes very long time to get that data. So before that data, we can check the accelerated stability testing. We can check the accelerated stability testing and we can find the shelf life. But in pharmaceutical company, they do both. First, they do the accelerated stability testing, and side by side, they will be also doing this real-time stability testing. So, from the accelerated stability analysis, they will find the shelf life quickly, and they will start to market the drug, or they will start to sell the drug, and side by side, they will also be doing the real-time stability test also. So for the accelerated stability analysis, the pharmaceutical preparation are subjected to high stress for certain period of time. Commonly used high stress input, temperature, humidity, light. अब high stress condition के use हुन्छ? एउटा high temperature, एउटा high humidity, एउटा high light. High condition मा चाहिँ ड्रगको डिग्रेडेसन प्याटर्न युज गरिन्छ. अब मैले चाहिँ यहाँ हाई टेम्परेचरको एक्जाम्पल दिएको छु है. हाई टेम्परेचरमा चाहिँ कसरी हाई टेम्परेचर युज गरेर कसरी स्टेबिलिटी टेस्ट गरिन्छ. जसरी As I mentioned earlier, we need RNS equation. यो RNS equation बाट चाहिँ हामीले चाहिँ ड्रग को reaction kinetics पत्ता लगाइन्छ. RNS equation gives the relationship between the rate constant and the temperature. If we can determine the value of the rate constant at room temperature, then we can calculate the shelf life. That is the remaining concentration of the product. यस्तो क्वेशन जस्तै अब generally हामीले चाहिँ रेट कन्स्टेन्ट के पत्ता लगाउनु पर्यो हैन, कुनै पनि ड्रग को डिग्रेडेसन प्याटर्न कस्तो छ हैन, त्यो ड्रग को डिग्रेडेसन प्याटर्न कस्तो छ, रेट काइनेटिक्स कस्तो छ भन्नाले के भ्यालु पत्ता लगाएपछि हामीले त्यसको चाहिँ रेट के भ्यालु पत्ता लगाएपछि त्यसको चाहिँ सेल्फ लाइफ भनेको त्यो ड्रग चाहिँ जस्तै 100% बाट 0% मा कन्भर्ट हुन कति टाइम लाग्छ भन्ने कुरा चाहिँ पत्ता लगाउन सकिन्छ हैन. सेल्फ लाइफ भनेको त्यही त हो, 100% बाट 0 मा हुन कति टाइम लाग्छ. अब त्यो चाहिँ सेल्फ लाइफ चाहिँ हामीले कुनमा पत्ता लगाउनु पर्यो, नर्मल टेम्परेचरमा, किनभने generally drug are stored in normal temperature 25 degree Celsius मा पत्ता लगाउनु पर्यो. 25 डिग्री सेल्सियस मा 100 बाट 0 मा कन्भर्ट हुन कति टाइम लाग्छ, त्यही हो सेल्फ लाइफ. त्यो चाहिँ केबाट पत्ता लगाउनु पर्यो, त्यसको लागि हामीले के चाहियो, के भ्यालु चाहियो, रिएक्सन कन्स्टेन्ट, रेट अर्डर काइनेटिक्स के चाहियो, केबाट के को भ्यालु चाहिँ केबाट पत्ता लगाउन सकिन्छ हामीले फ्रम द आरएनएस इक्वेसन. अब यो पत्ता लगाउन डिफरेन्ट स्टेप्स छ है. लेट्स सी द स्टेप. अब आरएनएस इक्वेसनलाई हामीले यसरी log k equal to log a minus ea upon 2.303 RT यो इक्वेसनमा हामीले चाहिँ कन्भर्ट गर्न सकिन्छ है. अब स्टेप्सहरु के छ, पहिला सुरु हामीले के गरिन्छ, त्यो फार्मास्युटिकल प्रिपरेसनलाई चाहिँ स्टोर गरिन्छ कस्तो कुन टेम्परेचरमा, हाई टेम्परेचरमा है. त्यो फार्मास्युटिकल टेम्परेचरले डिफरेन्ट हाई टेम्परेचर जस्तै 70 डिग्री सेल्सियस, 80 डिग्री सेल्सियस, 90 डिग्री सेल्सियस यस्ता डिफरेन्ट टेम्परेचरमा हामीले स्टोर गरिन्छ ताकि चाहिँ त्यसको डिग्रेडेसन चाहिँ भेरी फास्ट होस् हैन. And samples are withdrawn and the concentration are determined with the change in time intervals for each temperature. अब जस्तै 90 डिग्री सेल्सियसमा राखेको ड्रग को हामीले चाहिँ त्यसको डिग्रेडेसन प्याटर्न हेर्नलाई चाहिँ त्यसको सर्टेन टाइममा जस्तै 1 डे मा, 1 मन्थ मा हैन, 1 डे मा, 1 वीक मा, 15 डेज मा कसरी त्यसको डिग्रेडेसन प्याटर्न कसरी डिक्रिज भइरहेछ, त्यो ड्रग चाहिँ कन्सन्ट्रेसन चाहिँ हामीले निकालिन्छ. जस्तै अब 1 डे पछि त्यसको अमाउन्ट कति बाँकी रह्यो भन्ने कुरा हेरिन्छ, क्याल्कुलेट गरिन्छ. 10 डे मा कति बाँकी भयो त्यसको अमाउन्ट हेरिन्छ, 20 डे मा कति बाँकी भयो यसरी हेरिन्छ. जस्तै 90 डिग्री सेल्सियसलाई, 100 डिग्री सेल्सियसलाई, 70 डिग्री सेल्सियसलाई यस्तो डिफरेन्ट डिग्री सेल्सियसमा चाहिँ त्यसको चाहिँ कति डिग्रेड भइरहेछ हामीले क्याल्कुलेट गरिन्छ. अनि क्याल्कुलेट गरेर हामीले चाहिँ द अर्डर अफ रिएक्सन can be determined by plotting the function of concentration against time to get the straight line. For example, if you get the straight line while plotting ln a and time t, then the degradation is first order kinetics. अब मैले अघि भने के भने चाहिँ भने कुनै पनि ग्राफबाट चाहिँ हामीले चाहिँ त्यो ड्रग को डिग्रेडेसन प्याटर्न अथवा त्यो ड्रग को डिग्रेडेसन रिएक्सन फर्स्ट अर्डर हो कि सेकेन्ड अर्डर हो कि जीरो अर्डर हो कि हामीले निकाल्न सकिन्छ. ओके न. अब हामीले चाहिँ के गर्न पर्यो, त्यो डिफरेन्ट कन्सन्ट्रेसनमा हामीले चाहिँ त्यो ड्रग को डिग्रेडेसन प्याटर्न निकाल्न डिग्रेडेसन प्याटर्न हेर्न पर्यो. जस्तै अब त्यो ड्रग चाहिँ सर्टेन टाइममा कति कन्सन्ट्रेसन बाँकी छ त, कति कन्सन्ट्रेसन बाँकी छ भनेर यसरी प्लट गर्ने. जस्तै कन्सन्ट्रेसनलाई मात्र लिएर टाइमसँग प्लट गर्ने हैन. जस्तै एलएन र कन्सन्ट्रेसनलाई लिएर टाइममा प्लट गर्ने हैन, 1 बाइ कन्सन्ट्रेसनलाई लिएर टाइममा प्लट गर्ने हैन. यसरी प्लट गर्दाखेरि कुन चाहिँमा स्ट्रेट लाइन आयो त, जस्तै यो एलएन कन्सन्ट्रेसन बाइ टाइममा आयो कि ओन्ली कन्सन्ट्रेसन बाइ टाइममा आयो कि हेर्ने. अब हाम्रो चाहिँ यदि कन्सन्ट्रेसन र त्यो ड्रगको डिग्रेडेटिङ कन्सन्ट्रेसन र टाइम यसरी स्ट्रेट लाइनमा आयो भने चाहिँ यसमा नि आएन, यसमा नि आएन, यसमा स्ट्रेट लाइन आयो भने के बुझ्न पर्यो, हाम्रो त्यो ड्रग कसरी डिग्रेड हुँदो रहेछ, फर्स्ट अर्डर काइनेटिक्समा डिग्रेड हुँदो रहेछ भन्ने बुझ्नु पर्यो. ओके न. यदि यो फर्स्टमा नि स्ट्रेट लाइन आएन, सेकेन्डमा नि थर्डमा नि स्ट्रेट लाइन आएन, सेकेन्डमा स्ट्रेट लाइन आयो भने त्यो ड्रगको डिग्रेडेसन भनेको सेकेन्ड अर्डर रिएक्सन. यो किन इम्पोर्टेन्ट छ भने पहिला सुरु हामीले त्यो ड्रगको रेट कन्स्टेन्ट फर्स्ट अर्डर हो कि सेकेन्ड अर्डर हो भन्ने पत्ता लगाउनु पर्ने हुन्छ हैन. अब त्यो फर्स्ट अर्डर पत्ता लगाएपछि हामीले चाहिँ अब डिफरेन्ट अब हामीले चाहिँ अब सुरुमा चाहिँ 90 डिग्री सेल्सियसमा मात्रै सुरुमा 90 डिग्री सेल्सियस मात्रै राखेर हेर्यौं हैन 90 डिग्री सेल्सियसमा राखेर डिग्रेडेसन प्याटर्न हेरेपछि हामीले चाहिँ ल यो ड्रग चाहिँ फर्स्ट अर्डर काइनेटिक्स रहेछ भनेर पत्ता लाग्यो अब हामीले चाहिँ अब 90 डिग्री सेल्सियसमा के को भ्यालु फरक हुन्छ हो किन अब त्यस्तै हामीले त्यो के को भ्यालु चाहिँ 70 60 50 40 मा निकाल्छौं 70 60 40 50 मा त्यसको के को भ्यालु डिफरेन्ट हुन्छ हो किन K1 K2 K3 K4 K5 हुन्छ अब हामीले चाहिने के भ्यालु चाहिँ के हो यो के को भ्यालु चाहिँ 25 डिग्री सेल्सियसमा कति हुन्छ भन्ने कुरा हो किनभने 25 डिग्री सेल्सियसमा चाहिँ एकदम स्लो हुन्छ डाइरेक्ट हामीले चाहिँ त्यसलाई रिएक्सन गराएर एक्सपेरिमेन्टली निकाल्न एकदम गाह्रो छ के को भ्यालु 25 डिग्री सेल्सियसमा किनभने 25 डिग्री सेल्सियसमा ड्रगको डिग्रेडेसन एकदम स्लो हुन्छ त्यही भएर हामीले 70 60 40 90 हाई टेम्परेचरमा गरेको किनभने त्यसमा चाहिँ फास्ट रिएक्सन हुन्छ फास्ट के भ्यालु निकाल्न सकिन्छ अब के भ्यालु चाहिँ हामीले चाहिँ 25 डिग्री सेल्सियसमा कसरी निकाल्ने भने एक्स्ट्रापोलेट गरेर ग्राफबाट निकालिन्छ अब ग्राफबाट कसरी निकाल्ने भने सुरुमा पहिला सुरु अब लग के यो आरएनएस इक्वेसन भनेको इक्वेसन हो हैन यो इक्वेसनबाट हामीले चाहिँ के को भ्यालु चाहिँ हामीले चाहिँ 70 60 50 40 सबै थाहा छ हो हैन के को भ्यालु अब लग के y एक्सिसमा अनि यसको चाहिँ 1/t टेम्परेचर कुन टेम्परेचर लिएको त्यो लिएपछि हामीले चाहिँ यसरी एउटा स्ट्रेट लाइन आउँछ हाम्रो 70 डिग्री सेल्सियसमा 60 डिग्री सेल्सियसमा 50 डिग्री सेल्सियसमा 40 डिग्री सेल्सियसमा सर्टेन हामीले लग के को भ्यालु राखेर हामीले एउटा स्ट्रेट लाइन आउँछ हैन आरएनएस इक्वेसन किनभने यो इक्वेसन भनेको y = mx + c हो हैन यो इक्वेसन यो लग के र 1/t प्लटिङ गरेपछि एउटा सर्टेन लाइन आउँछ हैन यो सर्टेन लाइन आएपछि यो सर्टेन लाइनको हाम्रो चाहिँ के छ स्लोप पत्ता लगाउन सकिन्छ यसको स्लोप हामीले स्लोप क्याल्कुलेट निकाल्न सक्छौं हैन अनि स्लोप क्याल्कुलेट निकालेपछि अनि यसलाई चाहिँ यो ग्राफलाई एक्स्ट्रापोलेट गर्यो भने चाहिँ यसको 25 डिग्री सेल्सियसमा चाहिँ लग के को भ्यालु कति हो भन्ने निकाल्न सकिन्छ हो किन we can draw a graph by taking log k and on y axis and reciprocal of temperature on x axis यसरी 1/t by log k चाहिँ एउटा हामीले ग्राफ बनाउन सकिन्छ हैन this will give a straight line which will have the slope of magnitude equal to यति त्यो लाइनको स्लोप भनेकै यति हुन्छ किनभने m भनेको यो भयो हैन अनि यो लाइनको from this EA can be determined activation energy पनि निकाल्न सकिन्छ हामीले यसबाट है यसबाट एक्सप्रेसन पनि निकाल्न सकिन्छ the y intercept corresponds to log a from this the value of a can also be determined because यो log a भनेकै c intercept भनेकै log a हो हैन जसबाट चाहिँ हामीले चाहिँ a भनेको एक्सपेरिमेन्टली डिटरमाइन्ड भ्यालु मैले भन्थे हैन from the graph by extrapolating the value of log a can be determined for room temperature as well as for refrigerated temperature हैन रेफ्रिजरेटेड टेम्परेचर र रुम टेम्परेचरमा पनि लग के को भ्यालु पत्ता लगाउन सकिन्छ हैन रिएक्सन रेट कन्स्टेन्ट के निकालियो अब 25 डिग्री सेल्सियसमा के भ्यालु पत्ता लगाएपछि अब हाम्रो के छ त्यो के को भ्यालुलाई हामीले यहाँ राख्नु पर्यो अब हाम्रो यो फर्स्ट रिएक्सन हो भने अहिले हामीले भनिसक्यौं हैन एज अ एक्जाम्पल अब फर्स्ट अर्डर रिएक्सनमा चाहिँ के को भ्यालु पनि थाहा छ हामीलाई हैन के को भ्यालु पनि थाहा छ हामीले K25 को भ्यालु थाहा छ K25 को भ्यालु थाहा भएपछि अब हामीले टी निकाल्नु पर्यो टी भनेको सेल्फ लाइफ चाहिँ A0 भनेको इनिशियल कन्सन्ट्रेसन A भनेको चाहिँ फाइनल कन्सन्ट्रेसन चाहिँ 0 भनेपछि अब चाहिँ सेल्फ लाइफ चाहिँ A0 पनि थाहा छ A चाहिँ 0 अब रेट कन्स्टेन्ट पनि थाहा छ टी निकाल्नु पर्यो भनेपछि अब टी 25 डिग्री सेल्सियसमा कति हुन्छ त सेल्फ लाइफ भन्ने कुरा चाहिँ यसबाट निकाल्न सकिन्छ अल्टिमेटली टी 0 डिग्री सेल्सियसमा 0 डिग्री